the diversity on our planet is unique, and today I explained to my students that the chances of someone else being like them, genetically identical, is very slim. Now, subtract out identical twins, which I explained to them what that was. But the whole thing is, it's very unique. When you have 23 pairs of chromosomes, each one of them, these homologous chromosomes separate, and before they separate, they mix around and they allow for the shuffling of their genetic deck, so to speak. So what I want to do is a short video just kind of rehashing kind of what we're talking about. So genetic diversity, it's a review, and I'm not going to go into the details of gametogenesis, but I will talk about synapsis, crossing over, and how they can lead to diversity. Again, today's video is brought to you by Curious Moranland. Science literacy will make America great. That is our goal. So one of the things that my students copied down was they had a packet where I had this particular handout, and I like to look at these chromosomes. So I'm going to get them to think about certain aspects of this and how we can talk about diversity. Now, we're only looking at one homologous pair. So first, synapsis is the process that brings the homologous chromosomes together. Remember, they pair up so they can properly separate. So you see sister chromatids, but one's from the mother, one's from the father. So non-sister chromatids means exactly that. They're not from the same source. One's from the mother, one's from the father. But they will exchange pieces. So if you look over here, during prophase, which is the most important stage, prophase is where synapsis pairs up the chromosomes. You form that thing that we call a tetrad, but it also makes it possible for them to separate. We pair them so they can properly separate. If you don't do that, you're going to have what we call a non-disjunction, a chromosomal separation error that will cause some serious issues like Down syndrome, Klinefelter's, Jacob syndrome, Turner syndrome, a whole bunch of different ones that I'll go into later. So what I want my students to envision, so let's watch synapsis in action. Actually, this is going to show a karyotype, which is really what would result if synapsis occurred. I'm not going to label all of them, but here's the deal. When meiosis separates, it separates the paternal chromosome or the maternal chromosome. And here's what the result would look like. It's now a haploid cell. In matter of fact, it's a sperm cell, because over here you'll see here's the X, here's the Y. Well, and let's see if I can circle. It looks like this. Let's assume that this the left to right. So let's say this was the parental and this was the maternal that went. Here's the Y, so this child would be a male. Let's go back to this though. All right. So here's the deal. If this is the paternal and this is the maternal. Now this is what I tell my students. Envision this being like one of your parents' chromosomes, one of your parents' karyotypes. If we go back up to here, one of your parents' karyotypes, and when they get their chromosomes when they were born, they got one of their father's chromosomes and one of their mother's chromosomes. It's not a linear thing. It's not like paternal chromosome goes in and you send paternal to the offspring or you send maternal. It really is. If you look over here, during prophase 1, these chromosomes literally exchange pieces, and this is called crossing over. It's called crossing over. This is what further adds to the genetic detail. If you look at this, just separating like P from M, and P is paternal, M is maternal. If the homologous chromosomes just separated, they went in and left as they came in, that would create great diversity, because sometimes you'd be sending the paternal, sometimes you'd send the maternal. But what further adds to this is the exchange of the pieces. And I tell my students, say, like, for example, if this was your parent's chromosome, that means this paternal is your grandpa. This is your grandma. That means grandpa's chromosome, when it gets sent to you, if this is the chromosome that goes to the future gamete, that means it's not just 100% grandpa, it's grandpa and grandma, a little piece of grandma fused together. Or if this goes, it's grandma with a little bit of that. You genetically are unique. And this is one homologous pair. And this happens with all of our homologous pairs, with our 23 pairs. There's no crossing over between the X chromosome and the Y chromosome. So definitely with the autosomes, pairs 1 through 22. So watch a little look at this. This is just showing genes as we transition from learning about meiosis to seeing genes on a chromosome. And these letters over here, which might be cut off from the video, this is labeled. So this would be a trait, and then the letter represents it. We're going to call these an allele. So the trait might be red eye color. The allele would be the red allele or the white allele. Those are the variations, the possibilities. And you can ultimately link it back to a gene on a chromosome that came from both parents. Homologous chromosomes, same gene, same location, different source. This is, say that's from the mother, this is from the mother, and that would be, well, that's from the mother, that's from the father, because I've been going left to right. So, and then you can see how meiosis would separate those chromosomes, and then you have your, this is actually, let's erase that and try that again. I always tell my students, 4 cells. What you're looking at is this is one chromatid, this is another, this is another. They are uniquely different the way they come out from the way they went in. And we're talking about zygote formation and gametes and gametogenesis. Short little animation shows here's 4 chromosomes, two homologous pairs. They exchange a piece, and there we go. So I'm going to show you one more thing before we stop. All right, so I'm going to put the link to this video in the description if you want to watch this full animation. So when I hit next, I want you to look at the end result and the final result. They're going to show the homologous pairs separating, and they're also going to show them being sent to the next generation. So let's watch this. So I hit mute because this is from a website that's narrated. And again, I will put the description in the link. But take a look at this. So let's say, look at pairs 1 through 23. They've color-coded it to show here's homologous pair 1. Let's say this is the paternal and this is the maternal. Same thing with the mother in this particular case. So her paternal ones are, let's say those are here. And here's her paternal ones. And if I want to be technical, I probably should have called the bottom ones the paternal ones, because you see that Y for this particular guy. So look at the colors. This guy didn't send 100% green. He didn't send 100% red. She didn't send 100% blue or yellow. They sent a mixture. As long as each one of the chromosomes, so chromosome 1 from the father. Now, what they have done is, here's the father's chromosomes. Here's the mother's chromosomes. That's the way they lined it up. And so this is genetically unique. This is what he has. This combo he sent, like the first chromosome he sent was green, and then another green, and then red. The mother sent her 2 yellows, and then she sent some blues. What if they were to have another child? Of traits. So that's the narration of this. And again, this is from a website. So you heard a little bit of narration from the website. I was trying to edit that so you can actually just hear me talk over it. But so you see a unique combination of traits from the two parents. These kids are from the same sets of parents, but you'll notice they are genetically different than from each other and from their parents.